I like to trespass. And that means I don't feel bad at all in getting involved in areas where I know nothing. I just learn. I think that the intellectual pursuit should, orient you on what you have to learn next. So I practice philosophy without having a diploma. I practice working with, I'm not a lawyer or an economist, but I don't hesitate to work with them. So that is making bridges to arrive at some point that could be knowledge or an action. I'm very concerned about global warming, and I'm very concerned about endocrine disruptors and its consequences in the real world. And so I don't hesitate in getting into situations like testifying before Congress. And I did that in the States. That is my official address, Boston. So I did that in the Congress of the United States without many, good results. And here I testified 3 times before Assemblée Nationale. And there are laws in France that protect the children in particular, but everyone, about being exposed to certain endocrine disruptors. And so you go there and you have to learn something about economics and stuff like that in order to be able, not to the Congress or the Assembly, but to deal with situations where people would say, yes, but this is not economically possible. Well, I don't care in the sense of being able to be an economist, but you talk with people and you try to more or less get a perspective of the implications of your work. And here it's a different story, because here it's my own work that needs to be put into perspective. So I started, I don't know if you check the title, From Experimental Carcinogenesis to a Theory of Organisms and Back. What do I mean? I started life as a physician. As a physician, I already trespassed, because when I was a medical student, I decided to work in a laboratory, research laboratory, so I started doing science. And when I graduated, I tried to wear both hats, and it's very difficult to be a practicing physician and a scientist, because science cannot take priority over patients. I mean, you have to care about them. So I decided to finish my professional life and dedicate myself 100% to the pursuit of biology. So I started working on like every other scientist, biologist. Thank God, in my education, I had some exposure to philosophy. And that was useful at a given point in my career, and I'll tell you what and how. So the story is that a long time ago, I joined the lab as a postdoc of Carlos Sonnenschein, 1971. He had established the first estrogen-sensitive cell line. That is the time of the molecular biology revolution. Although I am a physician and I know a lot of physiology and all that, I was very much interested in in molecular biology, because we were brainwashed to believe that causation had to be studied at the lowest level of organization. So I was working on this type of thing. And when I joined his lab, because instead of working with a whole animal, there I could work with estrogen target cells, so it would be more reductionism. And I thought that that was a great thing. So according to the standard theory, the target cells proliferate when they are stimulated by hormones and remain quiescent in the absence of estrogens. This is what the people thought and wrote saying estrogens, this is the action of estrogens. However, the target cells proliferated equally well with or without estrogen in the conditions in which we put them in culture. But however, when injected into the animals, they proliferate exclusively if the animal was treated with estrogens. So you have a paradox. And so how do you solve the paradox? I mean, what do you do? So we started thinking how to deal with the paradox. And so this was the idea. I mean, how is that this happens? You have the cell, and you add estrogens or not. In culture, it didn't matter. You got proliferation. You got two cells. So we discovered, then we said, well, what is it? So what we did in a short, in a nutshell, is that we put serum in the culture medium. And the more serum you put in, the more inhibited the cells were. So we decided that what that meant is that there was a plasma-borne inhibitor. And when we added estrogen to this situation, now proliferation occurred. So the difference is in vivo, the animals are exposed to an inhibitor. In vitro, they are not unless you put serum into the concoction. But then if you go a little further, you say, well, okay, what is the point here? Because in microbiology, we studied bacteria and unicellular organisms like yeast. They only need nutrients to proliferate. So what is it that happens in metazoa where not all cells in the organism are proliferating? And why is that people that their natural state or constitutive state was quiescence, when in unicellular organisms and in plants is not the case, because you can take a leaf of any plant you wish and you rub the leaf like to make cells fall off into water plus a source of energy plus light plus nitrogen, and a plant is generated. Okay, so what is the difference with metazoa? And so we, at that point, is when two things happen. One, we decided to write a book after we have shown purified inhibitor and all that, and people were still thinking that quiescence was the natural state or constitutive state of metazoa. So we decided to write a book. That was my first sabbatical in France. And we decided as we were writing a book, we started to look at the bibliography, obviously, because people would say, it has been demonstrated that metazoan cells need growth factors to proliferate. Reference 1. So you go to reference 1, and it sends you to another reference 1 or 2, and you can go 50 years, and it hasn't been demonstrated. But then you say, well, what is demonstrated? So there is where the nebulous thing that at a point in your life you have been studying philosophy, however elementarily, comes to help you, and you start saying, well, here is not only a problem of science, it's a problem of epistemology. And so my next sabbatical was here over there at the École Normale Supérieure, Centre Cavaillés. I think I was the first locataire, together with another, I don't know. We say that in French. So the first person that spent the sabbatical there was, well, I was one, and the other a philosopher from Spain. And so this is how I started trespassing into philosophy. And then, so what is it? Default state is what we call it, just by analogy with computers, so that didactically people will understand better what we talk about, but in reality it's the unconstrained state, or the well, unrestricted, prokaryotes, unicellular eukaryotes, metaphyta, all those have proliferation and motility as the default state or the constitutive state. But metazoa, well, to us was also proliferation and motility. You have to explain how that is restrained by the multicellular organism. So our point was, if data are not theory-free, and there is no theory of organism, what needs to be done? And so, because really philosophers know that very well, but it's not the case with biologists. So a colleague of mine, Dan Dennett, a philosopher at Tufts as well, he wrote once, and his philosophy is not mine, but he said something that I used a lot because I have to come to France to find the type of philosophy that I was interested in, and that is another seminar probably. There is no such a thing as philosophy-free science. There is only science whose philosophical baggage is taken on board without examination. So that is a kind of norm for me. And I try to teach that to my students because really that needs to be done in a ship. You bring your preconceived ideas without examining. And that is a very serious problem that we have that got a little bit out of control with the molecular biology revolution, because at the time of the molecular biology revolution is the time of computers and computer science. And of course, the idea that the structure of DNA is complementary brought the idea of coding into biology. Okay, but that is another seminar. And people started to think that you could, first of all, that everything is computer-like. You know, in the old days, people used to equate organisms with machines. That is not new. You can go to the time of the car. So what changes the type of machine that you are comparing the organism with? And it's totally metaphorical to talk about programs in biology, but that, as I said, is another seminar. But I started to study all these things and realize which were the points of blockage where these people decided that inspired them to think that you are observing reality. And so to me, it's obvious that that is not the case, that if you were a tick, what you feel, your environment is heat and smell. So you will tend to feel the heat of an animal, a mammalian, and the odor of the sebaceous glands. And that is your environment. That is what you want. So it's not the same. Obviously, it's not that whether or not they have a brain, it's what they can perceive sort of determines what you can see or perceive. So therefore, objects are not directly observables. It's a theory, the one that defines what are your observables. And that I'm directly quoting Einstein. So, well, after the sabbatical, we published a book that is called The Society of Cells, that one was translated in French and last year in Spanish, and now it's going to be published in Italian, where we analyze this story about what is the default state and also what is cancer. And then after we did that, it was time for a next sabbatical, and I was very much interested in being more ambitious and propose the construction of a theory of organisms. And I applied to the Pascal chairs, and I became a Pascal chair here at École Normale Supérieure. And so I put a group. So my host was Giuseppe Longo of the Department of Informatics. And then Paul-Antoine Miquel is a philosopher at the Université de Toulouse. Maël Monteville is a mathematician, physicist, and biologist, which also has an incredibly good formation in philosophy. Mathieu Montio is a philosopher and theoretical biologist. Nicole Perret, a philosopher. Arnaud Pocheville, both a biologist and with a good philosophical base. Carlos Sonnenschein, a physician and biologist like me, and myself. So, and we work on this for a couple of years, and we continue working after we published issue in Progress in Biophysics and Molecular Biology in 2016. And actually, all this work, since the time I realized that objects are not directly accessible, all this trespassing into philosophy and becoming a theoretical biologist, of course, wasn't something that you can call a commentary or a hobby or whatever. This is my guide about how to run a laboratory and run experiments, okay? So it became that, and it's influencing everything I do. So let's move on. So we have to talk now on the roles of theories. And I think that I have sort of said that already, but you can have it here more in detail. Scientific theories provide organizing principles and construct objectivity by framing observations and experiments. So if objects are not, the objects that you're interested in studying are not directly accessible, then is the theory the one that determines what your objects of study are, because it's a big, big commitment. And if you are talking about Newtonian physics, atoms are not observables there. They don't exist in the framework of Newtonian physics. Okay, for example. So the choice of observables is a major theoretical commitment. So, for example, the decision to investigate the relationship between pressure, volume, and temperature independently of the microscopic structure of matter originated thermodynamics. So you make that decision. And so what we did is we have to think what was the way to move from physics to biology. And there is where Giuseppe Longo, that is my host at the ENS, play a central role, because for years he has been working on that issue together with another, he's a mathematician, with a physicist, François Bailly. And so one of the things that we need to do is to define a limit case, the default state, which would be like a principle of biological inertia. The awareness that biology is about change, and therefore two important aspects of physics do not apply to biology. One is a predetermined phase space, because obviously from the first thing that was alive, you cannot humans or crocodiles. I mean, as evolution took place, and actually the great evolutionary biologist Stephen Jay Gould said that if you replay the tape of life, now the metaphor doesn't go with you people, because nowadays you don't need a tape recorder that has a tape, right? So you don't have to rewind the tape in order to hear something that you taped. You didn't tape, you just recorded it. But 20 and 30 years ago, that wasn't the case. So therefore, he said, if you rewind the tape of life and you replay it, you won't get to the same point, because of the contingency of evolution, okay? So therefore, you have to know that. And that is very important, that that means you don't have a predetermined phase space. And invariance and symmetry conservation are not there. I mean, if you were a zygote at a given time. So there is breaking of symmetries in every time that you take a breath, more or less. And then the other thing is awareness that biological objects are normative agents. And that comes from a biologist, Bichat, and Canguilhem, and his predecessor, Nietzsche. So what we call a default state, that in physics, I mean classical physics, would be inertia. If no causes modify the properties of a physical object, the object conserves its properties. So, and this is a way of saying it by Giuseppe Longo, the default state is what happens when nothing is done to the intended object or system. So inertial movement is a limit case. So Galileo made intelligible all physical movements as special cases of it. By describing this default state, he could focus on the analysis of the forces constraining it. Okay, so that is one of the few things that started the scientific revolution as we know it today, to set that limit case. So we will move now to the principles for a theory of organisms. That is what I have been working on for a while now. And we'll talk about the first thing is, we identified three principles: the default state, variation, and organization. And I'm going to talk about that and how that illuminates the way we study biology. So I said the default state is what happens when nothing is done to the intended object or system. In the case of mechanics, as I said, we are talking about inertia and classical forces. In biology, the unconstrained state is the presence of nutrients, appropriate pH, and appropriate atmospheric pressure, etc., is proliferation with variation and motility. And this is rooted in the cell theory and in the notion that cells are agents that generate their own changing rules. And you can see it, you know, how paramecia will fight, will eat other living stuff for food, and so it will have some predators. And many times it escapes the predator because it takes the right decision without a brain, but decision nonetheless. So, but sometimes it's caught. So being normative means that you can make mistakes as well. So the default state corresponds to the very Darwinian idea of descent with modification on which selection operates. And it's really a non-conservation principle for phenotypes. So he made it explicit, okay, because living entities reproduce following a geometrical progression, but he didn't say it like that. These are his own words. There is no exception to the rule that every organic being naturally increases at so high a rate that if not destroyed, the Earth will soon be covered by the progeny of a single pair. Okay, so he had an intuition about this that we formalized, making it a principle for the theory of organisms, but that goes perfectly well with Darwinian theory because at a given point, every organism was a cell, a single cell. So our proposal has straightforward consequences on what requires an explanation in the sense of a theoretical cause. So the default state doesn't need to be explained. It's a principle, like inertia is a principle. To the contrary, a departure from the default state, that is cellular quiescence, restrained variation, lack of motility, requires a causal explanation. So you turn things around. Instead of being looking for growth factors, you now look for inhibitors, for example. Instead of thinking that invasion and metastasizing in the case of cancer is something that needs to be induced, you think, well, this is what happens when the tissue is not controlling well the cells that are inside, because they really know how to migrate, invade, and move somewhere else. The default state, proliferation with variation and motility, expresses agency and modifies the causal structure with respect to the theories of the inert. And this is also important because you cannot predict much regarding what a given organism will do. It will have a repertoire of behaviors, but because it's a normative agent, you cannot be certain about what it's going to do. It's not a computer program. Okay? So, and also, I mean, I should say that that also modifies the causal structure, of course, because it will learn, for example, of experience, and so it's part of maybe it's the only biological cause per se that the organism can decide things like I will proliferate, I will move, and in this direction. So the second principle is the principle of variation, that is a source of novelty. And that was postulated by Longo and Monteville, my collaborators. And so in analyzing differences between physical objects and biological objects, they created the concept of extended criticality. And I just quote, we posit that the dynamics of biological organisms are not just processes, but permanent, extended critical transitions, and thus symmetry changes. Within the limits of a relative structural stability, variability is at the core of these transitions. Okay? So the relentless change addressed by the principle of variation points to the major difference between the theories of the inert and those of the living. Because objects in physics are generic. So an atom is an atom is an atom, right? And universal gravitation applies to an apple and a planet. But here we are individuals. And individuals mean that we are somehow different and somewhat different. And we have to account on this difference in order to create a generality that is our creation as researchers. I mean, we try to find something in common, but in reality, that is alive is different from someone else that is alive. So we have to look at history, contextuality, variability of biological objects. Historicity is very important. We can get self-organizing structures like Bénard cells, and we can have flames, but we can create them any day of the week. Okay? Whereas organisms, we are not good at creating them. Any organism comes from a pre-existing organism for the time being, since this age that we don't fathom where the first organism appeared, that we do not know how that transition took place. Anyway, I want to show you some practical consequences of this variation. We say that development is non-identical iterations of morphogenetic processes, and that is what differs from some physicists that study development, thinking that, like in the lung, the bronchia divide in a given way. Well, the angles of division are not the same. There is a lot of variation. And I want to show you something here, but I don't want to block the light. So do you have a pointer? Does someone have a pointer? Okay. So I will break the rule and come in front of the of, so my shadow will appear there. This and these are mammary glands, okay, at day 15 of embryonal development. So at day 18, you have a tree. So the stem and all the branches. Now, those are the same strain of animals, and even sometimes I look at the right and left, and they are different. So you see, in the same animal, you can have this, this, or that. Nonetheless, as we were talking before, and in our study of endocrine disruptors, we found that one of them, at environmentally relevant doses, when given to animals this age, produce an effect. And that effect increases the propensity to cancer, and you can measure that effect on the morphology. So that means that that difference is circumscribed. The mean and standard deviation of whatever you're measuring will move here, okay? So you can do statistics, but you are, if you plot every point, you have a constellation that is much bigger than it will be an error of measurement, because the standard deviation of a measurement in, say, if we say in classical physics, is your measuring interval. Is due to that, not to the object that is generic. Okay. So I think I went in the opposite direction. And the third principle is the principle of organization, that is a source of stability. And that was brought to the team of the Pascal chair by Matteo Mosio. That comes from a Barilian and Rosenberg background. The principle of organization is the fundamental source of biological stability. And of course, this is based, if you want to go further, you can go to Kant and learn that to him causality in biology was circular and different, because the parts depend on the organism and vice versa. And so it's not that the organism is even the sum of the organs, it's just the causal loop between the organism and its parts through time. So this concatenation and dependency is the idea of closure of constraints as the means to achieve and maintain stability. And this was in the past applied to intracellular processes, and we went further and use it in in macro scales now. So Mosio and Monteville explore the concept of constraints being conserved at the time scale of the process being constrained. And this concept opens a point of entry for the mathematization of biology, because they consider that the constraint itself has a time duration that is much bigger than the process that is being constrained. For example, an enzyme is present at a given concentration, and even some molecules are replaced, you have that concentration of enzyme, whereas the product and the precursor and the product move in a very different, shorter scale. So now you can mathematize. So we exploited this notion by modeling mammary gland morphogenesis using the default state and its constraints. And if I have time, I'll show that. So the principle of organization says that processes are physical and chemical changes that occur in those systems. They involve the alteration, consumption, or production of entities, like chemical reactions and even morphogenesis. And constraints are entities which, while acting upon these processes, can be said to remain unaffected by them, at least under certain conditions. And here is the little diagram where constraints are represented by these squiggles. So these squiggles are controlling these processes, and these processes, because they are they are closed by these constraints, become interdependent. And then you have constraints that come from outside as well. And so that accounts for the relationship with environment. So, and this brings us to organogenesis. The default state results in tissue growth by generation of new cells and production of extracellular matrix. This process is constrained by physical determination involving flows and forces. Shape is the result of this default state. Constraints like gravitation and the constraints created by the living organism, like muscle tension. For example, if you look at a certain bone, for example, the tibia, it has a protrusion here. And people ask, is that something that is determined how? I mean, it comes from the bone, it comes from the muscle. So what they did is they gave a paralyzing agent to embryos of a certain age. And you see that if you do that, the tibia doesn't grow that protrusion. That is the contraction of the muscle that is pulling the matrix and creating that shape. Okay? So shape is the result of this default state with constraints that are created, not only gravitation, but those that are created by the living organism. And the ability of the organism to generate new constraints at each new iteration generates diversity, given that these iterations are not identical. So that brings us to the experiment that we did about morphogenesis. And I want to say that here we have some determinants of shape that we have studied. This is a component of the extracellular matrix, the collagen. And this is another component of the extracellular matrix that is matrigel, is an extract of basement membrane. So those fibers get decorated by these globular proteins. So cells behave differently in these two different types of matrix, to the point that we found out, this is mammary gland cells, that when you have a lot of extracellular matrix, like in the top left, this is 50% matrigel, constant collagen. The less collagen, so we have rounded structures called acini, like the ones that exist in the mammary gland of lactating animals. And as you decrease the concentration of matrigel, you start to see elongated structures, okay? So we wanted to know how this plays, I mean, how this happens. And so this is the basic description of the system. According to us, there is a biological component, the cells, and a physical component that is the fibers. So the cells constrain each other as they multiply, they exert forces on each other, but they also exert forces on the collagen, organizing it. The collagen transmits those forces, which in turn constrain proliferation and motility, particularly in a directional way. So what we did here, and this is Mael Montevilles, I mean, he's a mathematician, so I have to make a detour here. Turing, many years ago, made a distinction of what is that you do with computers. You can do two things. You can do modeling, that is mathematical modeling, and that is strictly based on principles. So if you are doing physics, it depends on the type of physics. You use the principles of that type of physics. Okay, perfect. That is modeling. But you can do something else that he called imitating or imitation. And so you use whatever ideas you have at hand. Oh, this looks like this equation, say Schrödinger equation. And then you apply that to a phenomenon that has absolutely nothing to do with it. So what is going on today with all this mathematical modeling, so-called mathematical modeling of biological systems, is imitation. It's not in reality modeling from principles. So Mael decided to use our experiments to model. And so he modeled based on the principle of, of the default state, cells that proliferate and move, and constraints imposed on them by the cells themselves and the constraints, and the collagen fibers. So here is the simulation, but it comes directly for the application of the principles of how a single cell in the upper left is forming a duct. And the yellow points here is the lumen of the duct. And that goes and depends on how the fibers are organizing. So these little things indicate the orientation of the fiber. And this is okay. Is it there? I cannot move it? Yeah. So if I am lucky, I can, no, I don't, yeah, I am lucky. So this is how an acinus is being formed, a rounded structure. Okay. And here is like how a duct is formed, the elongated thing. So when we play the photographs on time, we go back to the initial because we are observing for 5 days. By 3 hours, by looking at 3 hours, you know what it's going to be, if a duct or an acinus, because it's determined at that point. Those asymmetries are created at that point or not. And the acinus, what you have is not this directional formation of fibers, and therefore you end up with a rounded structure, as I showed you a minute ago. So the theoretical conclusions is that the default state restricts the field of possible hypotheses on the behavior of cells. Closure of constraints links the agency of cells with the structure of the collagen network, and thus is a major determinant of epithelial structure. And variation is highly simplified here because it's randomly generated by cells while they move and proliferate in a predefined phase space. Okay. What we learn from modeling. The model based on the default state and closure of constraints was sufficient to generate the bistable situation of two outcomes, rounded versus elongated. And inhibitory layers to constrain cell proliferation and motility was required in order to prevent the open-ended growth of the epithelial structures. And there is experimental evidence for this because I told you that we were working on the presence of an inhibitor that we purified. Closure of constraints is operative at the tissue level of organization. Okay. With this, I can move to what is a neoplasm. I don't know if I have enough time. Yeah? Okay. So neoplasm, or a cancer, is something that, as a pathologist friend of mine will say, well, he's a pathologist, and we all agree that the hallmark of neoplasia is altered tissue organization and excessive accumulation of cells. Neoplasms are diagnosed by pathologists and only by pathologists using light microscopes. I mean, the light microscope got to the top of resolution, et cetera, still in the 19th century. We have improvements, but not terribly enormous. So, okay, it's an old instrument, but can still be the one we use for cancer. And an old pathologist friend of mine, talking about the molecular biologists, he said, they would never recognize cancer if it enters this room, because you cannot look at genes and say this is cancer. You have to look at this. And what you see, this is a normal mammary gland, you see ducts that are of epithelial cells that are surrounded by a stroma that is made of fibroblasts, and further away there are those adipose cells. However, when it gets to be a cancer, you still have those epithelial structures, but they are convoluted. They are surrounded by a much thicker stroma than this, etc. And not only biology, science, because we also borrow from physics, for example, and chemistry. The somatic mutation theory of cancer, that is the one that has been dominant since 1914, the one that says that a carcinogenic agent acts in a single cell, producing mutations in this DNA, okay, that this is how it happens, and that those mutations will make this cell to proliferate without control. Okay. So it's based on the idea, that the heritability of the neoplastic phenotype is due to somatic mutations of DNA in a single cell. Well, we know that, for example, the phenotype of the liver and its inheritance doesn't depend on mutations. A liver cell begets a liver cell, okay? A pancreas cell begets a pancreas cell. Carcinogenesis is a cell-level phenomenon, and carcinogenesis is a problem of the control of cell proliferation. Those are the tenets, and that is what you learn when you go to school, because that is imprinted into your mind. But to us, and then of course the default state of metazoan cells is quiescence. That was the starting point where we started to not be in agreement with everyone and study and start to say, what are you postulating? So this prompted us to propose a new theory of cancer, that the metaphor we use is cancer is developing going awry. And we are reaching the conclusion soon, but anyway. So what do I want to say here is contextuality is the name of the game. The stroma determines the branching pattern of the epithelium. So here is in the case of the mammary gland, but also happens in other situations. So what you do is you take a fetal mammary gland and separate the stroma from the parenchyma. That is, the parenchyma is the epithelial component that is making these ducts that branch in this characteristic way. And the salivary gland does the same, but the branching is kind of star-like. So you dissect these components and recombine them. And when you do that and you wait, the mammary gland stroma makes the epithelium of the salivary gland form a structure that looks like a mammary duct. When you put the mammary epithelium into the salivary gland stroma, you get a stellar shape, okay? Star-like shape. So determination of the shape comes from the interaction with a particular type of stroma. And that is typical also, this is known even for longer, if you recombine the epithelium and the dermis in different points, like for example, you take a bird and you take the stroma of the foot with the epithelium of the wing, the skin that you get is full of scales like the one of the leg. If you take the epithelium of the wing, the stroma of the wing, and recombine it with the epithelium of the leg, you get beautiful long feathers. So in any case, the stroma is also typical of an organ. That is why the salivary gland stroma, to which people didn't pay attention, is the one that is determining the structure, even if the epithelium came from a mammary gland. But at the end, the function of the mammary gland is to produce milk, and that is produced by the epithelium. Okay? So we move to my next slide is kidney organogenesis, and that is just to show my point. This structure, the nephron, is formed by the mesonephric mesenchyme, that is a type of stroma, fetal stroma, and the Wolffian duct. When these two things interact, even you can do it in vitro, you get this, the glomerulus, And there are 10 different cell types that form that derive from this. Now, this doesn't happen like if you put the nephrogenic mesenchyme in a dish, it doesn't matter what you add to it, you will never get these 10 cell types. Because cellular differentiation is a consequence of tissue differentiation. Only when there is this proliferation and movement would these 10 different cell types form in a particular position. You understand this? That is not that cells form an organism. Cells are formed as the organism is being formed. So you cannot get to differentiate these types of cells unless they are building a glomerulus. And this is also brings to the concept of morphogenetic field. That is a piece of tissue in the embryo that if you take it away or put it in another embryo, if that was the determinant that makes this arm, now you put it here and you will get an arm here, okay? So it determines the formation of a structure. So what we propose is the tissue organization field theory of carcinogenesis that is based on development and the existence of morphogenetic fields that says when you put a carcinogenic noxa, by the way, most carcinogens are not mutagens, just in case, you know. So you put a carcinogen, and the carcinogen, what it's doing is affecting the reciprocal interactions between stroma and epithelium that determines the formation of an organ or tissue. When that happens, the constraints to proliferation and movement loosen, get lost, weaker, and as a consequence of that, the tissue would undergo changes like dysplasia, metaplasia, and carcinoma. So cancer is a problem at the level of the tissue level of organization. The target of carcinogens are tissues. Cancer is a problem of tissue organization comparable to organogenesis. And the default state of cells is proliferation with variation and motility. This means that instead of trying to figure out what makes cells move or metastasis and invasion, you should study the opposite. What makes cells not move, stay in place, okay, and proliferate less, okay? Because the default state, the principle doesn't need to be explained. It's what limits the manifestation of this limit principle. So, to us, mutations are an epiphenomenon. They have no bearing on cancer, and that is demonstrated by two situations, that there are cancers without mutations and that there are mutations without cancer, and also mutations of genes that people working on the frame of the somatic mutation theory call oncogenes. Like, for example, now that sequencing is so cheap, people got to sequence the cells of very old people and see that, for example, the esophagus is full of mutations and the tissue is perfectly normal in those oncogenes, and the same in any tissue, normal tissue that you want. So these two things are not consistent with the somatic mutation theory. And it's so ingrained that actually, this is a paper that Prado published. I mean, the first author is Rondeau. And actually, I showed that to Thomas. And I said, Thomas, you are great. You are saying that our theory explains better a bunch of things. But my God, you have a problem here. You put that the disruption of tissue architecture produces mutations. And then what is interesting is that they criticize us in the next sentence for putting mutations there. What's the point? And I said, well, precisely, it's no point. It's so ingrained that people cannot even conceive that you would say, they are an epiphenomenon. It doesn't count. But it's interesting that someone was criticizing us for it. He laughed a lot, but anyway, that is what happens. So we say, not really, they are an epiphenomenon. So, and oncogenes are expressed in normal tissues, as I said, so-called oncogenes. Which brings us to what I will answer someone's question about these cancers. I think that it came from there. So we're talking about cancer. What happens? Well, here you have at higher magnification the normal duct. And here you have a duct that is a carcinoma in situ. Carcinoma in situ is defined by, it becomes bigger, and is full of bridges of these epithelial cells. So the epithelial cells grow towards the lumen and fill it with these bridges. So that decreases functionality. So a cancer is less functional. Why? Because if you have blocks, the function of a duct is to let things go through, right? So milk in this case. So that will be blocked. So the function is hindered. However, this is more complex than this because here you have topologically, here we have one lumen, here you have 1, 2, 3, 1, 2, 3, 4, 5, okay? So more complex, less organized because organization means function. So in order to see which one of the two theories goes better with the outcomes that we get in real life, say, we ask the question, which is the tissue, the target of carcinogenesis? So we separated mammary gland, epithelium, and stroma, and we expose each one of them independently to a carcinogen. A carcinogen that has a half-life of 20 minutes. Then we rinsed several times and wait 5 days before we recombine that. So there comes the experiment. You recombine the thing, and then you wait quite a long time. So those are the controls. That means just the rats, you inject them with the carcinogen and with the vehicle, and you get tumors only when you inject the carcinogen. But if you recombine, here is both have vehicles, so nothing happens, negative control. Here you have the epithelium with the carcinogen and the stroma with vehicle, nothing happens. If you expose both of them to carcinogen, you get tumors. And if you expose only the stroma, you get tumors as well. So this is consistent with the tissue organization field theory and inconsistent with the somatic mutation theory. And this is just to show you one of the animals that had a recombinant. So you get real tumors, and then you look histologically, and they are for real. The other interesting thing is that our theory predicts that tumors are reversible because mutations are forever, but tissue organization can be remodeled. And this is the experiment we did. We took several cancers, and we obtained cells from them. And then here is a complicated thing if you are not a biologist. But the mammary gland, when the animal is very small, the epithelium is very small. So you can cut it. You go to the mammary gland, make an incision, and cut it, and then take that piece and look at the microscope that your margins are clean. In other words, that you didn't leave any epithelium behind. After you do that, you let the animals heal, okay? So at 11 days of age, at 10 days of age, we do this thing that is called clearing of the mammary glands. You remove the epithelium. And then you recombine with the cells that you obtain from the tumor. And I'll explain the gist of the experiment later. So we injected those cells at 24 days of age, 52, 80, and 150. And then animals that were pregnant and had babies and lactated twice. And we asked what the outcome will be. Now, why did we do that? Because if you inject that carcinogen, you get 100% tumors at 24 and 52 days of age. But if you inject here, you almost get only 5% instead of 100%. So we thought, well, okay, since we believe that the stroma is so important and the reciprocal interactions, we will see a difference if we inject at different points. So the hypothesis is that here you will have lots of tumors, and here we'll have very little, and here very little. So we ran it, and effectively, the primary tumor is there. In the animals injected at 27 days of age, is more anaplastic, that is less differentiated even, and all of them got tumors. If you inject it at 52 days of age, very anaplastic tumor coming from here, okay? And here I don't put tumors because they were not tumors, so what I show you is the mammary gland itself in whole mount, and what you see is that these cells that once belonged to a cancer, develop all these beautiful ducts. So yes, cancer is reversible and depends on the reciprocal interaction of stroma and epithelium. And this is not the single paper. There are approximately, in liver, melanoma, hepatocarcinoma, melanoma, mammary gland, and a bunch of other tumors. In total, I found about 55 references where you can control cancer. Moreover, there are many cancers that regress spontaneously in children. And what you see, okay, you don't see anything because the children are cured, but say you operate because of an appendicitis. So you operate here, and you see a big ganglion, and so you take it because in the clinical history it says that the guy had a neuroblastoma, and it's a perfectly, and that it was localized, perfectly normal big thing. So it normalizes, not that it was attacked by the immune system and destroyed. So what we are talking is normalization, not destruction. Okay? So we learned that a group of normal cells in the wrong morphogenetic field may generate a process akin to carcinogenesis in animals. And carcinogenesis in an animal may be prevented or reversed when tumor cells are put in contact with normal cells, of the different components, so epithelial cells with stromal cells, and etc. I can explain more if you want, but I think I'm taking too long already. Conclusions, and we get to the end. We have proposed principles for a theory of organisms that provide an appropriate framework to the study of development and cancer. This theory framework provides means to perform mathematical modeling from principles rather than using applications from engineering and physics. This theory approach explains carcinogenesis as a problem of tissue organization. The metaphor is morphogenesis going awry. From the tissue organization field theory perspective, cancer is a process that can be healed by reversal to normalcy, by reorganization of the constraints. So I have to thank my group, the organism group, and also the people that put money in what we have to do. This is very important today, particularly UNS Foundation and Gouvernement d'Île-de-France, because that was an incredible opportunity to do theory in one of the best places where you can do theory in the world. That is this beloved institution of ours. So, and I thank my scientific partner, Dr. Sonnenschein, and all these people that one way or another have participated in this research that was done in several places in this world, Boston, University of Ulster, Oxford University, and Mass General Hospital. And thank you very much. I will be more than happy if you have questions that I